Okay, so this talk is mostly just going to be an introduction to pharmacological fMRI. Mostly, I'm just going to describe what it is and some of the confounding factors about what the difficulty of doing fMRI when you put a drug into somebody's body, and how to deal with that, so that you're trying to make sure that you get the actual neural signal. And hopefully give you a couple of types of examples that are in the literature right now. So pharmacological fMRI is really basically just an fMRI experiment where you give the person a drug. And mostly you're looking for the modulation of, instead of just giving them a task where you're just looking at the modulation of their resting state data with the task, or the brain activity with respect to in response to the task. You're looking for the changes that you get either in the resting state networks, if you were at Katie's talk on Monday, and changes that you end up getting that you can measure with tasks, or just actually looking at the pharmacokinetics of the drug uptake in the person's brain. So one of the key things to remember is that the bold signal that we use in fMRI is actually a function of a couple of different things. So that if you give somebody a drug, you're going to be changing these things. So you have to be a little bit careful that you're actually measuring neural change and not changes in blood flow, blood volume, or just metabolic changes. So the point of pharmacological fMRI, just to be abundantly clear, is to demonstrate the effect of the drug. Did the drug actually manage to get into the brain? Because not all of the drugs actually penetrate the blood-brain barrier. It might also help in choosing a dose. A lot of doses are actually given in terms of dose per weight, and that may not actually be how that affects the brain. You want to maybe also be able to choose whether or not the drug is actually working early on in drug development, and I'll talk about that in a second. You want to actually choose neural targets for the drug to make sure that you actually are using a drug which is addressing the network or the circuit issue that you have. And mostly it's also just a scientific tool for modulating brains, just like giving somebody a task. So in terms of the drug development process, if you are sort of not familiar with it, it actually takes a really long time and a lot of money in order to actually develop a drug. So if you start with, if you start and look at where the preclinical research is, you have 10,000 targets in terms of drugs that you might potentially have. Over the number of years here, it's about 20, I think, you end up with one drug at the end of this whole process. And if you look at the number of drugs that are actually end up being approved, it's maybe 22 per year, which is not really very many. So this sort of drives one of the reasons why you might want to actually insert imaging in any one of these stages, because you'll be able to actually verify whether some of these 10,000 drugs works earlier in the process, instead of spending all of your money on these clinical trials later on. And you can see that people are spending a lot of money to buy drugs, so developing new drugs is actually a very interesting thing to do. But since it's so expensive, a lot of the companies have stopped actually developing CNS drugs because not many of them actually make it to market. So a lot of these companies are losing money and they're losing interest in actually developing drugs. So imaging can maybe help by showing up in the preclinical development of the drug to actually make sure that the drug is actually looking at the target. These drugs are typically developed in preclinical models like mice and rodents, and maybe if you're looking at something like depression, mouse depression may not be the same thing as human depression, and so that may be one of the reasons that the drugs that they think work don't end up working when they actually try to translate it into the clinical trial phase. So if you actually then look at the various parts of the drug, the clinical trial itself, in terms of the clinical trials are separated into 4 different phases. In the earliest phases, basically, you can actually make sure that the target is being engaged. In the secondary phases, you can make sure that the drug is actually working. And in the later phases, you can actually make sure that you're actually targeting the drug to the right group of people. So, as I briefly mentioned, one of the problems of doing drugs that actually affect the brain is not all of them actually manage to get into the brain itself because of the blood-brain barrier. Using something like fMRI, which is a non-invasive technique, you actually end up being able to actually measure this repeatedly and for a longer period of time to actually make sure that you're affecting the parts of the brain that you hope to. You can actually measure this uptake in the brain, which you can't do with most other techniques. And peripheral blood measures may not be ideal in terms of actually figuring out if you've actually engaged your neural targets. Another thing that it might help with is the subjective response to drugs. People, when you give them drugs, they are the ones that report whether they feel better, for example, in depression. But they might be having something called the placebo response. So the imaging may be a more objective way of actually seeing whether or not the person is actually responding to the drug, and separate people who respond to the drug from those that don't respond. Because you may end up with, you get this confounding effect of the person saying, oh, hey, I feel something, when really you didn't actually give them a drug at all. So if you actually have proper imaging targets, you can actually tell the difference between whether they actually are responding to the drug or whether they're not really actually responding at all. When it comes to looking at study design, in terms of looking at drugs, you can, if you have this drug uptake curve, there are really a couple of different phases that you're interested in. The baseline effect, and also the peak effect, and anything post the drug. And there are different ways of breaking that up, just the same way as you would look at either task at any of these stages. In particular, you might be really interested in actually just the uptake of the curve and just no task at all, no sampling, just measuring the whole thing. One of the problems there is just that certain drugs like antidepressants, they take several weeks to act, so you really can't put the person in the scanner for for 2 weeks at a time in order to figure out what the uptake is. So this really only works well for drugs that have a rapid uptake. What you end up really looking at, for the most part, are actually just samples at various points in time. Say this is not just minutes, it could be over a set of weeks. And then you actually have the confounding factor of the fact that the person's mental state is not the same at every scan. So let me just show you a couple of different examples of things that exist in the literature. So here is an example of a trial where they gave healthy participants a drug called midazolam, and it's an anti-epileptic, if I've got that right. You've got resting state networks that they're highlighting here. So this is the motor network. So there's motor cortex that's lighting up here, and this is without the drug. And so when they give them the sedative, basically you can see very little difference, but there's a difference in the anterior cingulate, or the cingulate here. Same thing in the default mode. There's a little bit of a change in the PCC seed. If you don't find that super convincing, this example is a little bit better in terms of looking at the effect of pain response. So here they're basically giving them a bit of a painful stimulus on the back of their hand, and you can see that the stimulus is identified by each of the dashed lines, so you can see that the person is responding pretty nicely, or you can at least see the response to each of the stimuli. And in the second panel, you see the same thing, except in the gray region, they're actually giving them a drug called remifentanil, and you can see that the response to the drug is considerably reduced. So this is one of the nice things that you might be able to do, is titrate the dose so that they just get rid of these responses. As an example of something that you can look at where you just give an acute drug, this is where they do an acute injection of nicotine, and you can see in various regions of the brain that the uptake curve is pretty well visible to the naked eye, and that way you can actually see what the plasma, and that it matches pretty nicely with the peripheral measure in the blood plasma as well. the next thing that you can also do is the same kind of thing where you're actually looking at the bold response and trying to actually match it to the perceived pain. So there's no reason that the bold response necessarily actually measures that the actual pharmacokinetic response to the drug, just because it may actually be mixed up, as I'll talk about later, with some of the effects in terms of, like, it might, the drug might actually change blood volume, blood flow. So in this experiment, they were basically just making sure that the bold response that they were getting actually matches the perceived pain that the person was actually getting. So in this pretty nice review of the resting state literature, this person looked at, there's about 100 different papers out there that are looking at testing drugs. You can see that there are a lot of different drugs that people are trying. Some of them are therapeutic, some of them are, more recreational. And about 75% of these are actually straight fMRI experiments. And they're looking at just trying to figure out what the drug is doing. In some cases, trying to actually see if there's a particular response to this drug. And I think this is still sort of in the development phases. All right, so moving right along, let's look at what there are possible confounding factors to actually trying to measure a drug response. Right, so let's first talk about the cognitive effects. Unlike sort of when you give somebody a task where you actually can see their response and see if they actually got the question right or wrong, there's something called the placebo effect, which most people should be familiar with. And in fact, the placebo effect, if you're looking at antidepressants, the placebo effect is actually increasing over time. So studies that were done many years ago, say 10 or 20 years ago, had actually lower placebo rates than studies that are done now. And the reason for this increase isn't really well known. It could be just that people are, more familiar with medications actually supposed to be helping them, so they, think that they actually get a response. There are actually a couple of studies that have been done. This one most notably by Helen Mayberg, where they tried to actually isolate the brain responses that are actually responsible for the placebo response. And this sort of cartoon actually demonstrates that a lot of the regions that are responsible for the placebo response that are shown in the ones that are common are shown in the filled-in circles, and then the ones that are specific to the like antidepressant response in this case, where the drug is actually being in effect, are clear. So you can actually see that a lot of the drug response and the placebo response networks are actually the same, and only a very few of them are actually specific to the drug. So imaging might be one way that you can actually see if the person is actually having a placebo response rather than a drug response. The other thing that people do, and there's a lot of effort that's put into this, is to try to design their study such that they end up randomizing people either to getting drug or placebo so they can actually tease out the differences. And there are a couple of different ways of doing that. There's parallel trials where you end up giving drug and placebo to different people. Crossover trials, which you give this, the person goes from one arm of the trial where they get a drug and then they cross over to the placebo arm, where so the same person gets drug and the same person also gets placebo. Of course, this takes a really long time because you have to wait for the drug to wash out, and then and then you can give them the placebo. So people then try to do things like matched pair trials, where you get participants that are approximately the same and matched in a lot of different ways, and you just randomize them to drug and and placebo, and you try to do it that way. There are a lot of considerations to these kinds of study designs. Some people, in terms of whether you give open label or randomized, double-blind tasks, or where neither the participant nor the actual researcher actually knows which drug is being given. And a lot of these design considerations are what you actually want to get out of the trial. You also basically want to figure out how many subjects you want. Some of these designs require more subjects than others, and also how long it is that you want to spend, doing or collecting the data. Some of the examples from our group, you'll see that a lot of these are actually crossover trials. Some of them are actually not. Some of them are just open label trials. And all of these have to do with ketamine. But we do try to actually incorporate imaging at many different time points in order to actually try to figure out whether, where the drug effects come from. In some cases, we're trying to look at the acute and the relapse. Sometimes we're looking at the actual uptake. And we also try to involve multiple imaging modalities. So a little bit more about difficulties in terms of acquisition of the data. If you're looking at, say, drug uptake in the brain, these are really slow changes, and they're on the same scale as drift artifacts in fMRI. So one of the solutions to that is to actually look at, get multi-echo data. So let me explain to you a little bit why imaging slow stimuli doesn't work very well. In this case, you've got a panel which gives you bold data on one side, and you can see this is a motor task which alternates at about 30 seconds on and 30 seconds off. You end up being able to see the motor task really nicely. As you increase the time that the person is spending doing the task and not doing the task to a minute, and so forth, you can actually see that eventually you can no longer actually localize the bold signal after a while. But what they did was they acquired simultaneous ASL, and though the signal was a little bit weaker in ASL, you can see that the person was valiantly doing the task the entire time. So why is it that bold? So basically the slow drift artifacts are sort of canceling out the actual task that the person is doing. It's obscuring the response. So one of the things that people will end up trying to do is basically model this, all of these slow drifts. You can see in this particular paper what they did was they basically took a set of different models and tried to fit it, and basically chose the best model, and they were like, oh, this is the best model, this is what our data looks like. But what happens if you don't actually know what that model is? You might be removing some of the signal that you're actually looking for. So these drifts end up having, being down in the region that we're looking for. Most of the time, people can actually choose their task frequency to be outside of it. And so, how can we get around doing that? So one of the ways to do that, I'll explain to you in a second, and just to remind you, if you didn't go to some of the previous lectures, everybody is reasonably aware at this point that bold is actually not a direct measurement of the neural activity. It's very indirect. And what we do measure is a single point in time in looking at the difference between the oxy and deoxyhemoglobin signal, right? So we get this one single measurement. It turns out that bold has this nice property that it scales with the time that we actually measure it at. So that time is actually called TE. So normally we get a single image per picture that we get for a scan. And so instead of doing that, we can actually get 3 brain images. And if we do that, we end up with images that have slightly different contrasts because they're sampling this contrast curve at different locations. And if we then look at the time series from one particular region, you'll see that the time series is actually scaling. And we can actually take advantage of this scaling property to plot basically the signal versus the TE of the image that we took, and you'll see that it scales nicely. So if it scales, then and we can use this scaling property to actually choose components that are ones that we're actually interested in looking at. And so this is the principle behind multi-echo denoising, and I think there's a lecture from Javier later on in this series which goes into this in more detail. But just to recap it briefly, as I was saying, if you have a signal that scales, that's a bold signal and you want to keep it. If it doesn't scale, it's not. So you can actually rank components that, say, come out of an ICA if you've done multi-echo acquisition in terms of how well they fit this scaling model. If they fit the model well, like the default mode network that you see here, then you want to keep it. And if it's looks like this motion artifact here, which has a low amount of fit to this model, or doesn't fit the model, you can throw it out. So let me demonstrate to you that that actually does work using sort of a surrogate task, which has a bunch of different ways of scaling the contrast. So this is kind of like, something that's decreasing in terms of, the drug is ebbing away. But instead of using a drug, I'm actually using just a flickering checkerboard, which scales with contrast. So you should get a similar curve in the visual cortex. So if you actually then look at the data, this is just the raw data that comes out, you can see that a blocked task, for example, looks pretty much like a block. You can kind of see that it's there, but this ramp task, you can't see at all, right? So now if you do some standard fMRI processing, since you can see the block before, you can see the block is it's better resolved. But if you do the normal pre-processing, you still can't see this curve anymore. Now with multi-echo denoising, it's convenient that you can still see the block design. It's not removing the task signal, but now you can at least see that it's positive up here and negative down here. So now you're starting to be able to see this curve. And part of the reason, if you look at the components that were rejected, or the non-bold components, you can see that it's a positive going trend. So a positive going trend plus the negative going trend gives you this flat line that you see here, which is something that you can't actually remove natively with the single echo data. So multi-echo data might be one way to actually get at looking directly at the pharmaceutical uptake. And just to show you that it actually works, the multi-echo data shows that the stimulus is actually localized to the visual cortex, and it's the only one of the different types of pre-processing that actually shows you that it works in the case of the ramp task. Losing my cursor. Okay, so now if we're looking at some more signal confounds. One of the things that happens if you give somebody a drug is it also changes their physiological processes like heart rate, blood pressure, and so on. And if you went to the talk by Dan Hanwerker earlier, he probably explained to you, and I think he did a good job of explaining why doing some of those peripheral measurements is very important, and correcting for those changes is important in order to actually get proper, say, resting state networks, et cetera. And that's the same that's the same kind of thing that we have to do here. Because instead of measuring, we want to actually measure the neural changes from the drug. So, in the control case, if you have this kind of signal, for example, you want to make sure that you're finding a neural response and not just a vascular response, right? So this is the case that we're hoping to see. Of course, and the same thing when you give the person a drug. So if you see this decrease in response, it's purely due to neural response and not due to vascular response. But what might actually happen is that this particular patient might just have a negative vascular response, and so it looks like they have no response at all, but they do have this actual vascular response here. So when you give them a drug, you don't see any change at all. But that's not actually, we want to be measuring this particular condition and sort of not account for these vascular responses. So fMRI itself can't actually necessarily detect these things. You can use multi-echo, like I said before, but one of the better things to do is maybe actually use something like MEG or EEG, where you're directly measuring the neural signals, right? One problem with doing that is that if you're measuring MEG, for example, that's a separate session. Maybe you're interested in the better spatial localization that you can get from fMRI, so you can maybe try and combine the two things together. So just looking at EEG signal origins, as I said, those are directly neural. So if we do measure these things at the same time as fMRI, which we can actually do with a little bit of difficulty, then you have an extra confirmation that the drug changes that you see are actually real. So if this is such a nice thing to do because it combines basically the nice temporal accuracy of EEG and the nice spatial accuracy of fMRI, or spatial resolution, why don't more people do it? I mean, if you have a look at the data if you just count the number of publications, lots of publications in resting state fMRI, some publications in resting state EEG, but not so many in the combination of the two of them. And I think there's a lecture coming up by, I think, Pete Bulfes, who's going to go into EEG fMRI in detail, so you can go to that if you're interested in it. Part of the reason is that it's a pretty complicated setup. You end up not just having the scanner, but a couple of other computers to actually be able to do the EEG acquisition. And just to explain a little bit why it's a little bit more complicated, is the signal that you're interested in is on the order of 10 to 150 microvolts. If you put the EEG in the MR environment, you end up with scanner artifacts, one that comes from the scanner imaging gradients itself, which are on the order of millivolts. These are pretty easy to remove because they're they happen regularly. The one that is or not very difficult, but at least more difficult to involve is what's called the ballistic cardiographic artifact, which is on the same order of magnitude as the signal that you're looking for, and that comes from the heartbeats of the person interacting with the magnetic field, and that's why it's a little bit more difficult to remove, because you have to actually identify the time points where the person is actually having a heartbeat. And there are also other artifacts coming from just the person being alive as well. So one example, though, that this does really work if you do simultaneous EEG and fMRI to have a look at drug effects, is this one, where they did two different tasks, one which was really easy, which is just a simple visual task. You can see that it localizes pretty nicely to the visual cortex. And then they looked at the visually evoked potentials from EEG. So those are coming from the visual cortex as well. But you can see that the caffeine doesn't actually change the visually evoked potentials at all in this particular case. Now, looking at the auditory oddball task, the fMRI localizes the caffeine effect to be in the frontal lobe, same thing as with the EEG. If you look at the evoked response here, they're actually showing a difference in the caffeine versus the non-caffeine states. Which is an interesting application of the two. So in this particular case, caffeine affects tasks that are more difficult, doesn't seem to change anything in terms of an easy task. You can do the same thing in terms of trying to validate the simple example that I showed you before. So this is just to remind you of the block task versus the ramp task. Conveniently, a flickering checkerboard is also nicely visible as a frequency peak in EEG. So you can actually do the same thing to look at the time course in EEG by looking at just the task frequency. So if you look at the task frequency, you can see that it's a visual signal just in the back of the head. Keep clicking on the thing. And if you look in the temporal domain, you can actually see that the task is actually faithfully represented in EEG, as well as the multi-echo denoise data. So lastly, if you were sort of paying attention earlier, I showed the slide where I had the comparison of Boole data and ASL data, and you might wonder, well, if ASL is so great in terms of picking up these slow changes, why wouldn't you just use ASL to actually measure your pharmacological changes? ASL, I mean, as you can see here, gives you slightly less signal than Boole does. It is still, but it does actually pick up these differences. And the reason being is that it's a difference between two images. And so since you're getting the difference between the two images, you're not getting that slow drift artifact because you keep taking differences between image pairs. And so this is a slide that shows you what is being done in ASL. I think Lilith is giving a talk on perfusion imaging later. ASL is nice because it gives you a signal which is directly proportional to the cerebral blood flow, which is a quantitative, more quantitative measure of what's happening in the brain. Boole tends to give you signal that comes mostly from places that are close to the veins, and this gives you more signal that's coming from the periphery. That's considered to be not necessarily more neural, but it's more quantitative. Nicely, you can actually do these things together in terms of interleaving the two scans. And somebody actually did this. They've actually given the subjects MDMA, where they see generally a decrease in various regions, notably the amygdala. And then they also do a similar analysis in resting state, where they also note differences or decreases in amygdalar connectivity. So there are actually a lot of different analysis types that you can actually do, and mostly, a lot of the different techniques that people use, any of the fancy ones like graph theory, et cetera, can be used and applied to all of this stuff as well. And just in summary, pharmacological fMRI can have benefit in terms of mapping drug effects in the human brain, but it's still quite challenging and very much in development. And there are different ways of trying to deal with the various confounds that you might have in terms of the bold signal, either by combining it with other things like EEG or using slightly different sequences. And with that, I'll just conclude and answer any questions that you might have. Which I'm assuming there's none, so that's fine. And then I can just let you guys go so you can get on with your day. Yeah.
Is there a way to combat the strengthening of that placebo effect that you mentioned?
I mean, there are a lot of people who are looking at, design of studies, and I'm not really sure that. I mean, I think at the last conference I was at, they had a lot of good luck by just telling everybody they were getting drug to start with, and then seeing what happened afterwards. But I think that's sort of an open area of research in terms of how to figure out how to convince people. I mean, that's just a perceptual thing. Some people are just more susceptible to that than others.
So you said EEG fMRI is more accurate generally, but just the trouble is the artifacts that it creates.
Oh, sorry, I just remembered that I was supposed to actually repeat the questions. So the question is about whether EEG is more accurate, and that one of the problems is that it just has more artifacts than just standard EEG or fMRI. It's not so much that it's more accurate, it gives you information from two different modalities, right? So you end up, like EEG has really good temporal resolution, but really bad spatial resolution, because you either have 256, electrodes, and then you have to back project to get the actual spatial resolution. fMRI, on the other hand, has decent temporal resolution, very good spatial resolution. So it's really being able to combine the data from both of the modalities to give you better characterization of the signal that you're looking at. And the artifacts are a problem. But one of the things you're doing, if you're doing EEG and fMRI at the same time, is if you can find a biomarker or something that actually identifies your drug, you can then translate it into the clinic. It's a lot easier to do EEG than it is to do fMRI, insofar as it's a lot less expensive. So if you're doing simultaneous experiments and you can validate something in EEG, you can then take it and do experiments just in EEG a lot cheaper and a lot faster outside the EMR environment. So if there's no more questions, you can either come up and ask me, or you can just